I'll try and, I know at least one person has a headache, so I'll try not to speak too loudly. So the structure of phospholipids, I'm not going to get too much into detail there, but I want to use this time wisely. So I will say that ultimately you should have had something that looks like this. I don't know what's going on out there, but something that has two lipids hanging off it. There should be, you might have added the nitrogen or not, but certainly I'd expect to see a phosphate, right? You might have put a phosphorus with 4 oxygens. Phosphate has 4 oxygens on it and it has an extra negative because, well, let's just leave it at that. But we draw this, and often it's drawn in biology with a P with a circle on it. All right. I think somebody already went, but if there's a pass up there, you can use it. You can go. So that should be your phospholipid. When you're drawing phospholipids in membranes, we generally don't do all that. We just draw a circle. And the wiggly line, every time there's an end to the line, there's a carbon. So there's 1 carbon, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. Does that make sense? There's a carbon. Everywhere you go look, there's carbon. Carbon-based life form. So at this point also, you should be able to know that's hydrophilic, correct? And that you got the hydrophobic ends, they do that. And that you got a bunch of these that are laying out in a circle. And yeah, that's correct. And in the first iteration of this is going to be a micelle, where you have kind of this going on. And we talked about this being a detergent. This is what would happen initially, where again, you have the hydrophilic side. It's either hydrophilic could mean charged or it can be polar, either one, but they're hydrophilic. They love water, both charges. And you have the hydrophobic, which are just lipid, which are just hydrocarbons or carbon and hydrogen, all laid out. And this is a sphere. So to be fair, it would look something like this if you were looking at it. If you found it, you looked underneath some kind of supermarket microscope, you'd find the lipids would just be a ball. Just be a big ball of hydrophilic heads, sorry. You wouldn't see the inside unless you cut it with a knife in half, and then this is what you see. So this is the cut version. Cut it, and that sphere turns into this. So that's not good for life because it does not allow there to be a water inside. Water is the solvent for life, so we need to be able to have water inside. So we need a bipolar layer. That's where this comes in, a bilipid layer. And so obviously, or at least I hope it's obvious, this continues and you have that going on. And, I don't know how that happened. And, they form a sphere. It's a sphere, but it's hard for me to draw a sphere, so you have to live with it. It's supposed to be, and so I'm just gonna, instead of continuing this, I'm just gonna kind of draw a circle here. And just know that you have this, you know what, let me just do that. And you have this internal lipid layer that I'm just going to go ahead and just color in like this. It's kind of the main idea here is that you have these, and of course these are all this would be yellow because they're all lipid. Correct? And you could have, obviously you had more time, so you could have drawn it. I don't want to waste your time trying to draw this out here, but that's what it would look like. And why is a bilipid layer, right? Why is that such an important invention?
It keeps foreign objects out of the cell.
That's right. It separates outside from inside. That's a big deal because before this happened, by the way, so did this. The micelle did as well. But the difference is that with the bilipid, you have an environment inside, a water soluble. There's water as the solvent inside. Okay, water as the solvent inside. And there's an environment that's different than the outside. There's an environment that's different from the outside, and that's a big deal. Where the micelle, you had an environment that was different from the outside, but you didn't have water as a solvent. So now you couldn't do some of the things you need to do for life. All right, so let's go back and look at the questions. The questions what? The first question was draw the phospholipid structure, draw it. How did phospholipids sort? We did that in the micelle, and then we talked. We said it's necessary not to have just a micelle, but to have a bilipid layer. And then what are the characteristics of life, and how does the cell accomplish what's necessary for that? Well, the bilipid layer is important, but then we have to think about how does that accomplish the characteristics of life. But what are the characteristics of life? And to look at characteristics of life, you're really talking about one, organization. I'll take that even one step further because the way life organizes is something called compartmentalization. Big word. I believe that I spelled that right. Did I spell it right? Correct? Please remember that. Okay, because you love to remember when I misspell things. So compartmentalization is how we organize. Okay? That's how life organizes. I'm going to skip some space because I want to add to it because I'm going to revise my notes. So I'm going to make sure I have plenty of space. That was shade. That was throwing shade at a lot of you.
When we were at Case, we saw the lecture board, and it's really bad. The handwriting is really bad.
Yeah, you need to, when you get a chance to visit colleges and you see these professors and what they do,
It was like a bunch
not everyone's had the training that you've had at this school. And they become PhDs and they become teachers. PhDs are not necessarily teachers. Yeah. You go, you're good. I'll post this video. They're not necessarily teachers. They're really fantastic. They have knowledge that I don't have because they're PhDs and they specialize in their field. It's important that you get the opportunity to learn from them, but understand at that level, you're learning from them. They're not necessarily teaching. So you have to have the skills to pick out what you can get from them. And that goes for when you're in medical school too. A good physician is a good teacher. That's going to be your primary job as a physician, unless you're a surgeon. Your primary job is going to be to teach people what the heck to do. Like you're going to have to convince them to stop eating salt. So you have to, you're going to have, or sugar or whatever, you have to convince them to change their lives. which means you have to educate them. That's going to be a really big part of your career as a physician. And like you can have the answer, but if they're not going to do it, if they're not going to comply, forget about it. So organization, what's the second one? Give me a second one.
Growth.
Reproduction and growth, I heard. Reproduction.
But it's different, separate.
And the third one here is growth. Response to stimuli. Homeostasis, all good ones. Every book has a different list of them. Some will call organization, some will call compartmentalization. There's one that's missing, it's important, probably one a very big important one.
Yeah, energy.
Okay, we can add use of energy. Maybe we can. Could we add it somewhere else? Well, let's just use energy. That's fine. Use of energy.
Oh, adaptation. Yeah.
Oh, evolution?
Adaptation.
Seven, adaptation.
What about evolution? Or is that the same?
That's adaptation.
Is it?
Yeah. Oh, that.
C/evolution. Right.
If you mean by adapt, they change on the go, then you're probably talking about responding to stimuli.
Can you say adaptation? Like, can you connect it to neuroplasticity? Like, because you're basically talking about a biological being.
Yeah, neuroplasticity is important. Could you add that to
Could you involve it with adaptation?
not if you're talking about in the concept of evolution. When we talk about adaptation, usually, and it's confusing, I hate this, but when they'll use adaptation, sometimes they're talking about responding to stimuli, which would be more of your neuroplasticity, the idea that you can develop into different tissues depending on where you are in this developmental process. Development, you can even add development, right? So we said reproduction and growth. You can say growth is one thing. That means you're growing more muscle, let's say, or you're growing taller, versus another one, which is development, right? Like you were very different when you were in the womb, right? We talked about that. As a male, we're very cognizant of the fact that we were all female in the womb, and that we developed into males before we came out. We turned out various genes and we changed. But we changed a lot. Like we had tails in the womb, we had gill slits in the womb, we looked like worms in the womb, we looked like tadpoles in the womb. We had web fingers in the womb, web toes. All this stuff changed as we grew and developed, and we continue to develop now. You're developing right now, and I'm developing. We're continuously changing, continuously developing. So this one's actually very much tied to responding to stimuli, right? And also adaptation and that / evolution. So it depends on using the word, but usually adaptation is associated with evolution, where neuroplasticity is dealing with development. All right. So in looking at these words, there's 8 of them. Let's take where I'm going to focus on for this discussion. You can connect everything to this, to these characteristics. It's a pretty comprehensive list. We could probably break it down and connect and kind of integrate a couple of these, but we can leave that out. We can talk about reproduction. Reproduction can happen in various ways. You can have, There's a reproduction. The cell cycle can be used in growth or reproduction, right? Yeah. With reproduction, there's going to be kind of two big categories. You have asexual and sexual. And when we're talking about asexual, you can talk about binary fission, which happens mostly in single-celled organisms. Well, it happens in single-celled organisms, period. That's the only time. Prokaryotes, specifically prokaryotes, actually. You call them and I call them bacteria. So that's where binary fission is happening. They do something, they have a cell cycle. It doesn't look a lot like our cell cycle, eukaryotic cell cycles. There is asexual eukaryotic. There is asexual division for eukaryotes. And eukaryotes use a process called, they have a nucleus, so they have to divide that. They have a nucleus, and so they call it mitosis. And the cell cycle. So that's the cell cycle you guys are talking about that happens in eukaryotes, and that includes a process called mitosis. So asexual division happens through mitosis in a eukaryote. Sexual division is meiosis. And so sexual division happens in eukaryotes. And we call that meiosis. Right? There are other types though. So in eukaryotes you can also have other types. I'm not going to go into it, but let's say other types.
Wait, meiosis is when it does...
Meiosis is when you take and you're going to basically, there's a whole process. We'll go over it. We'll remind you of it. It's no different from what you learned in ninth grade. It's exactly the same meiosis. No additions. I did everything that you need to know from AP biology meiosis. You already did in ninth grade. But we'll go over it again. But there are other types. For instance, there are single-celled organisms called Tetrahymena that have 6 different sexes. And they have more than one nucleus. And they have self-replicating DNA. And they have all kinds of interesting genetics and like they do so many interesting things that are different from what we would talk about in a regular biology class. And you can learn about that as you move on in college. But let's just put it all under other types, right? Sexual division in eukaryotes is usually done through meiosis. But even in meiosis, there are kind of small changes. We as humans, we will double our DNA. We will do our separation of sister chromatids differently in meiosis, so that we get this mixing up of the DNA. There's crossing over. Then we half the DNA again, and we end up with 4 cells that all have half the DNA, as we call them gametes. But in some plants, we'll double the DNA and keep the double DNA, and you'll have one generation of plants, like corn, could have 100 chromosomes, and the next generation could have less than that. And they do all kinds of crazy genetics that we don't need to get into, but just so you know, not everything's as simple as we're presenting. Response to stimuli, I think that's easy. Poke it and it moves, right? Food and it eats. Stimuli, give it a stimulus, it does something. Of course, that does all these. You can't. It's hard to separate them because you do a response to stimuli. You're going to need to have energy for that, right? Right. Homeostasis, that's important, and of course, the cell membrane is important for that, and adaptation, evolution. So now let's go back and look at how the membrane is involved in all that. So when you're talking about even the simplest thing, like eukaryote versus prokaryote. Okay, when I learned this back in the day, my professor taught us there were two spellings. One was European, the other American. So it's not true. I found out later it's not true, but it's too late. Now it's in my head. It's called fossilized knowledge. And now I'm constantly getting it confused in my head because it always goes back to my teacher teaching me one thing that was wrong. And so I'm always trying to make sure I correct my mistakes. But understand, once we get things fossilized in our heads, it's always hard to undo. So, prokaryote versus eukaryote. So, eukaryotic, prokaryotic, the very essence of the difference between these two big groups, right? You agree they're big groups. The very difference to do the big groups is because of the membrane, how much membrane you have. And in fact, how much compartment do you have? How much organization do you have? Now I'm not going to draw the phospholipid bilayer, I'm just going to draw a line. We use blue lines so that's all there is to that. So prokaryote has only one membrane, one major membrane. There's no membrane-bound organelles. I'll never understand what happened. What happened? It disappeared. Did you see it disappeared, right? One. Maybe. I must be doing something. No. Membrane-bound organelles. No membrane-bound organelles. And that includes, of course, no nucleus. Now, this sounds like just facts, and in honors biology, they were exactly that. They were tested. And here you do have DNA, and it's sitting there. I'm going to draw DNA in yellow. No, you know what? Let me do it in green, so it's different. But it's a circular DNA. So you have little ones called plasmids, and they do have ribosomes. I'll do the ribosomes in red. They have ribosomes, and that's about it. Those are prokaryotes. Simple, one compartment. One compartment. Everything happens in the same space, as one space. The simplest, right? You have the micelle, and then now you do have a bilipid layer. You do have proteins. Some prokaryotes do have one kind of other structure known as a flagella. All right. Some don't. Some are circular. Some are cosi. So they're little rods. E. coli are rods. Coccus are circular.
The little ones that come out and they help the neuro.
The flagella. This is the flagella. Some do have flagella, but they only have one compartment. Everything's done in there. They eat, they poop, they do all their processing all in this space. Eukaryotes are much more organized. Yeah, some do. All male humans have cells that have flagella. We call them sperm. You do have cilia. Remember the difference between cilia? Yeah. So, I don't know of any prokaryotes that have cilia. Ciliates are generally protists. They're eukaryotes.
They're the ones with the little things coming out, right?
Right. Cilia. Let me draw cilia. You have cilia. You have cells with cilia in your throat. You ever get a tickle in your throat? And you ever feel like you're kind of working it out? You're coughing, you're trying to work out the phlegm?
It moves it around, right? Like
When you cough, you have a cold, and you feel it in your lungs, and you feel it. You're kind of working it up your throat. How do you think you get it up out of your lungs? The cilia. They're literally moving it up.
It's on the sides.
It's all in your throats. It's your esophagus, or not esophagus, your trachea is a cylinder, and in the cylinder you have these little, we call them fingers, you can call them like little fingers, they're doing this all day. And you move the phlegm out from your lungs out to the mouth where you can get it out of you, or nose, or whatever it is. And they look like this, but there are single-celled organisms like the Tetrahymena thermophila that I just mentioned that are single-celled. They do have a nucleus. They do have a membrane-bound organelle. They have a nucleolus. In fact, they have a couple nuclei. They have 2 nuclei. They do have an endoplasmic reticulum. They do have a Golgi body. They do have different cell organelles. I'm kind of simply drawing it. But they also have these little things like this, little cilia, like almost little rowboats that help them run around. They run around, they eat. These things eat bacteria. They're much bigger than bacteria. I drew this bacteria very big, but they're actually very small. If I were can I shrink it? Bacteria is very tiny. In fact, they're very tiny. I can't. Okay. Okay. You're not going to work with me? Very tiny. Very tiny. And the cilia will come around and go and eat the bacteria, all right? So the prokaryotes are very small compared to the eukaryotes. Now, when we talk about our eukaryotes, the whole point here is that these are all spaces within spaces within spaces. And if you think about it, all your organelles in your body, and when we let's look at a human eukaryotic cell. Let's go zoom in and draw a giant human eukaryotic. We have a nucleus, a nucleolus. Those are 2 different spaces. Membrane bound. The nucleus is membrane bound. Nucleolus is not. But so you have this space here. That is a membrane, right? This is membrane number 1. All the blue lines are membranes. Let me do it. Blue equals membrane. That's phospholipid membrane. Got it? All right. So connected to this, you have an endoplasmic reticulum. The red is ribosome. That's right. So you have a small, rough, and a smooth ER. Oops, I put it in the wrong order. Rough and small ER, and smooth ER. And I'm drawing a line, but remember, this has 3 dimensions. It goes down too. It surrounds the whole nucleus. This is all it's thick. There's spaces in here. This is a bilipid layer. So so if I would zoom in, what you would see is that this is all a space. There are spaces inside this membrane. It's creating this compartment where different chemistry can happen. See, that's the key, is if you have different spaces, you can do different chemistry. This could be acidic, this could be basic, this could be This could have a lot of oxygen in it. This could have no oxygen in it. Like you can separate stuff. And that lets you do different kinds of chemistry in different spaces. You have another membrane called the Golgi body. And that packages it. And it creates, and from the smooth ER, again, the rough and smooth ER goes all the way around. I'm drawing it very simply. But from the rough and smooth ER, you can have the vesicles. These are vesicles that transport, right? Vesicles. Also membranes. And then you have the mitochondria, which has double membranes. The powerhouse of the cell. I hate that term, you know that, but whatever. It has double membrane. Where ATP is produced from sugar or from pyruvate, but the key is that you have two sides. So now you can create a concentration gradient. Now you can have power. Because here's the thing, what do you need to have a battery? What do you need to have to create a battery? What do you need?
Positive and negative.
Yeah, that's right. You need a positive end and a negative end. You need an anode and a cathode, and that means you have to have two different spaces. Every battery has something dividing the positives and the negatives inside it. You have to be able to separate them, otherwise the chemistry it doesn't go, it doesn't work. So you produce two different spaces, and that's how mitochondria do what they do. If it's a plant or a photosynthesizing single-celled eukaryote, some kind of algae, then it's also going to have a chloroplast, which has double membranes, the thylakoid membranes inside and the membrane around the chloroplast. And I should do this blue. To do photosynthesis, I should do the internal membrane green. That way everybody understands it. You have ribosomes, they're not membrane bound, but you do have these other things called lysosomes. They have digestive enzymes. So this is a really great example of having different stuff in different cells. Here you have these horrible little enzymes. They're horrible little enzymes. I'm going to put, it's Halloween, so I'm just going to put little fangs on them. They will destroy, they are made to break down every polymer.
They look like serpents.
That's what they are, pretty much. They're really like...
Oh, they're kind of cute.
Are they? You don't, when you die, this is what turns you into your dead body into gelatin.
Oh.
So when we dig you out, if assuming you have no way of drying out, when we open you up, you go from this dead stiff, they call them stiff sometimes, because you're stiff, because you're not moving. As you break down, you turn into gelatin because of these things called lysosomes.
So they break out.
They break down. Their membrane breaks down as soon as they get out. They don't actually eat. They just catalyze, right? They speed up the reaction of breaking down. So they'll break down proteins, lipids, everything. And why do you have these things laying about? Well, because you're going to have something called endocytosis, because you need to eat, because you're trying to maintain homeostasis, so therefore you need energy, right? So all this, see how related this all is? It's a whole system. That means it's completely related. So in here you have some food. Maybe it's what's left you have to digest it, obviously break it down so it gets absorbed. And when you absorb food, it doesn't just come into your cell, it has to go into, it creates a vacuole which brings us to another organelle called a food vacuole.
What is a vacuum do?
It just holds stuff. A vacuole means empty. A food vacuole is not empty. It has what?
Stuff.
Food. So this is a food vacuole. So it's just a space that has food in it. How do you break that food down? These two fuse. The lysosome fuses with this one. The lysosomes get to the food. They get deactivated. They themselves are broken down. We can break down everything in ourselves. So everything that is built can be unbuilt. You have to be able to get the waste out. So you have this. You produce waste, and what the kind of waste you produce is ammonia, NH3. That's really bad. NH4, I think is what it is. Really, I can't remember. 4 or 3. Ammonia, bad. It kills you. We use it to wash floors, kill bacteria. So your body turns that into urea. And that concentrates into urine in your bladder. So you need to transport this in your blood. You can't have ammonia in your blood, you'll die. So you turn into urea salt. It's a salt. And in your kidneys are really good at managing salt. It takes the urea, puts it into the bladder, and it takes and balances the salt in the water, pulls out as much water as it can, and you go from this little bit of urea in the blood to a lot of urea in the urine, and so we call it urine, which is water mixed with a lot of this salt. So that's also why if you eat a lot of protein, especially animal protein, you can get something called gout, which is urea crystals forming in your blood, and the crystals of urea are sharp. They're sharp like needles. So you got all these needles floating around in your blood, and that gets stuck and gives you a lot of pain and swelling. Very painful. My brother had it. It was horrible. So you have to get urea out. How do you get the urea out? Well, you produce the waste. So you have another vacuole, a waste vacuole, right? Same idea, just a membrane produced by what? Probably the Golgi body, right? Maybe endoplasmic reticulum, but whatever. Yeah, but this is called a waste vacuole. What do you have inside? You have urea, salt. I don't know what color to make that.
Do purple.
Do we have purple? I think we use purple.
It says custom color palette.
All right, let's do another one. What color is this here?
Purple.
All right, there you go.
Sure.
You happy?
Yeah.
All right, so here you have urea crystals. Oh, I think this is, for some reason, I'll pass these at 100%.
Worked.
So you have urea crystals.
Except purple. Kind of pink. Is it same as the inside?
Huh? Yeah, it's similar, but not the same. But let's just go ahead and change it to something else
It's
as
a
I
little
can.
darker.
The blue is yellow. We haven't used yellow.
We didn't use yellow?
We haven't used yellow.
Oh, no yellow. Okay, so we'll make it yellow. So these urea, yellow makes sense. Urea yellow. Why didn't I think of that? Urea salt. So we take the ammonia and we make it into urea, and that goes off and goes and bonds with and fuses with the cell membrane. And what happens is, instead of coming in, this is going out. It fuses and out goes the urea into the bloodstream.
Into the blood.
So this is going out, where this stuff is going in, and the other one's going out. This is endocytosis. Yes, this is Latin, and this is where Latin actually comes in handy. Exocytosis. I lose Latin every day when I'm breaking down words. That's what I did for a semester in college, is breaking down words. Latin can be very useful. The exercises you do in Latin, you can use it to understand the rest of the planet. So just know that. So in any case, these are all the membranes that are inside a eukaryote. Every space is being used for a different purpose, a different chemistry. This one's breaking things down. This one's building proteins. This one's packaging them.
That's transporting.
This is transporting in. This is transporting out. This is making ATP. This is making ATP and sugar. Yeah.
So, like, does this all have this much space, or is it jam-packed?
Has enough space. They also are kind of jammed up if you look at it. But no, there's a lot of space. These are all very tiny things. In a plant cell, you have one organelle making sugar, the other one's eating sugar. Two different spaces, right? Mitochondria break sugar down, chloroplasts make it. So in a plant cell, you're doing both. Chugga, chugga, chugga. That's why plants don't need us. We need plants. They can make their own sugar and eat it. We need them. We should be very thankful plants are around. I thank my plants every day. Thank you. Yes?
Is that ammonia or ammonia?
That's a good question. I think this is ammonium, NH3 is ammonia. I don't remember which one's made. I'm sorry, I don't.
NH3 is ammonia.
Ammonia, and then NH4 ammonium. I'm not sure. You can look up the chemistry of it. I don't remember, to be honest. Yeah. The difference is one's NH has 3 hydrogens, the other's 4. One's plus charged. The other, this is ammonium. It's a plus. It has to do with the chemistry of it. I just honestly don't remember, so I'm just going to let it go.
Function, though?
Huh?
Do they have the same function?
They're just a breakdown of, and when you eat food, when you process the thing, what your waste, your body's waste, is ammonia. Now, carbon dioxide is also part of that waste, but that goes out of your cell through diffusion, and you breathe it out. But the protein gets broken down. You need nitrogen. The nitrogen needs to come out somehow. Your nitrogenous waste comes out as urea. Yeah.
Where is ammonia processed if it's so dangerous?
We almost immediately turn into urea. Everywhere in the cell, there's enzymes everywhere ready to take any ammonia you get and turn into urea.
Is there like something that could stop that from happening?
You'd be dead. You wouldn't be born if you did not take ammonia, nitrogenous waste, and turn it to urea. Very quickly the cell would stop existing. It wouldn't be able to handle it. It's very deadly stuff. That's why we use it to clean floors, because it kills bacteria. It's also stinky, but that's another story. Are we good? So all this is about membranes, right? All this information, I should make it bigger, I suppose.
Yeah.
How do I select it though? That's the question.
Don't you
I do that, but then it doubles it every time.
just zoom?
Crop it.
Yeah. Hey, why are you opening it?
You should have said yes to me. I'll kick you. Wow.
So all that, so when we're dealing with this in a eukaryote, all that's because of membranes. And what is that? What is this? Let's look at these processes. Let's look at it in terms of these characteristics of life. Well, where's the processing of energy happening? Well, that's the mitochondria and the chloroplast, right? And where's the, oh my goodness. Please help me. Stop. No. No.
What is going on? What are you doing?
It's trying to grab, I'm trying to move it, zoom out, and it's grabbing everything. So where's the reproduction stuff? The mitosis, meiosis is going to be happening in the nucleus. Organizations, every one of these organelles helping organize stuff, compartmentalization, right? Growth is going to be doubling this and growing this, so you need endocytosis, bringing in food. You need exocytosis, taking out waste. You need homeostasis is happening. Well, everything here together keeps balance. You need to get rid of waste and bring in food, otherwise you can't get energy. You need energy to keep balance. If you have to respond to the environment, so as you're bringing in food, you have to respond to that by making more enzymes, by moving the vacuoles around. Remember those two little feet and those two little proteins that move across the microtubules? You need to drag the vesicle from one side of the cell to the other, right? Do you remember that video? I can pull it up. It always freaks me out though every time I do. I don't like it.
Let's see it.
Me no like it. Yeah, it's because it's Halloween. You want to scare me, that's all. So I don't like that video. It scares me.
Why?
You don't remember it?
No, I don't.
Oh, I'll show it.